Hey everyone, welcome back to the show. Today my guest is Krispn Given. Krispn is a professor at Purdue University and he has a program there called the Mite Biter program, which we're going to hear all about. He's also the co-owner of a business called Apis Engineering, which produces high quality insemination equipment that is made in the US. Krispn, welcome to the show.
Thanks for having me.
Now, Krispn, in doing research for this episode, I've concluded that you've spent a fair bit of time looking down a microscope.
Yes, that's certainly true. Yes.
Yeah, just reading through when you were doing the assessment of the mite biters, the amazing amount of time you would have had to spend counting the damage and all that kind of stuff is just incredible. Well done.
Yeah, thank you. Unfortunately, the last few years, the last 15, 20, about, I guess, 16 years now, I've been fortunate to have an undergrad to help. So when we bring the mites into the laboratory, they're able to look at all of them. But in the beginning, I did do that work myself. I was looking at mites for hours, distinguishing which mice were damaged, etc. It's very tedious work. Yes.
So do you want to tell listeners what a mite biter is?
Okay, so mite biting, this is a trait that disseminates basically from grooming behavior. So you have honeybees, they don't have a lot of mite resistant mechanisms because they haven't been dealing with varroa destructor that long. So, but one of them is grooming and mite biting. So mite biting is simply what it is. It's where the bee will grab the mite and bite it, and in the best case scenario, they will not only bite it, but they'll remove legs or chew into the shell of the mite, the idiosoma. So when we see more damage in the shell, for example, maybe that reflects a higher response to selection.
We've talked about this a little bit on the podcast before in the episode with Brent and Michael Simmons, and he called it allogrooming. Is it the same thing?
Well, there's different mechanisms. It's part of that allogrooming. So once the grooming behavior, they have what we call auto grooming also. So individuals, and then you have bees that respond to the grooming. And so the grooming behavior, we found that bees that groom more mites, for example, in our studies that we've conducted here, we found that bees that groom themselves more also chew more mites. So there's a correlation there. So that was exciting to see. A few years ago, we published a paper, one of our recent papers, where we don't know anything about mite biting and very little about the grooming. And we had a hunch that there was a gene responsible, Nixon, but we weren't sure. And then we kind of amplified that selection process and looked at it closer, and we found that indeed Nixon was correlated to grooming behavior. So bees that groom more also chew more mites. So that was really nice that we found in that study, because there hasn't been a lot of research done on as far as grooming or mite biting. There's a lot we don't know. So we only know of one gene that's related to grooming behavior, as I speak, and we don't know if it's recessive or dominant or as VSH bees, for example, there's a few genes that are responsible. So it's we call it additive type trait.
And so grooming behavior is not associated with VSH.
No, it's isolated. You can kind of think of them as VSH is kind of an indirect attack on varroa destructor, or the grooming and mite biting is a direct attack. So with VSH, years ago, they called VSH, varroa sensitive hygiene, SMR, suppression of mite reproduction. And then they changed the name because they discovered that the bees were detecting a smell, some anomaly in the infected pupae, or where a mite family was a founder's mice goes in, and she's successful at reproducing, and then they're able to detect this distress signal basically from the pupae. So that's causing the uncapping. And then you have another bee come in and he'll scan the pupae with her antennae. And in some cases, even though they've initiated uncapping, one bee did. So that's one gene for a trait. And another one would be a bee coming by and scanning and verifying if it really is infected. So in some cases, that bee that's scanning will determine, hey, this pupae's fine, and then they'll recap it. So then we have another bee, or bees come along and they recap the cell. But if it is indeed infected, then with VSH, then they will have what we call undertakers, and they will remove the infected pupae and cannibalize it, and eat parts of it, and then other parts they fly out of the colony.
Mhm. So in your program, are you only selecting for grooming behavior, or are you also trying to introduce VSH into the lines?
Yeah, that's a good question. So years ago, when we started, we just looked at colonies that grew the fewest mites. So we would put a sticky board in for 48 hours and remove it, look at them, and we just selected from the fewest mites. But also in the beginning, we did incorporate SMR back then they were called. So we got some of the SMR bees, the VSH, and then we also got Russians that we incorporated into our breeding program because Russians are good groomers already. They're better groomers because they've been associated with this parasite longer in the Primorsky region of Russia. So we got that, and then we got many diverse sources, survivor sources, and then we got our commercial beekeeper here in Indiana who works with over 3,000 colonies. We got stock from them. They're stationary colonies that do not go to the California almonds. And so we just selected from that through the years. And so, yes, we did get some VSH bees. We got a couple queens we got one from John Harbaugh, and then we got three. We got two from Tom Glenn, who used to be a breeder in California who's retired.
So from the research that I've done, I've discovered that you started off with 5% of your colonies that were mite biters, and now you're up to 50%. Is that true?
Yeah. So when we started looking at this trait in 2007, we were just around 5%. And then we've managed around 2015, we got it up to around 50%. And it's plateaued. That's about where we're at. So it's an incredible trajectory of, selection. So what's this doing is it's eliminating the mite population. It's eliminating mites from reproducing. So it's helping the honeybees because it's not so much what you see, it's what you don't see. It's causing a lot of these problems, which are unfortunately going to affect your country now. It's the viruses associated with the mites. So they're parasites. So you can think of the varroa mite as a dirty hypodermic needle. So it's the viruses, like the deformed wing virus, for example, that they're vectoring this, causing colonies to die because it compromises the immune system of the individual bees and then also the whole society, the whole colony bees. It's like going to a rock concert and somebody that has a flu, so eventually it gets passed through each individual.
Mm-hmm.
So it's a devastating impact the varroa destructor is having, and it's the most devastating because of the viruses it vectors.
Mm-hmm. You want to tell listeners how you assess a colony as a mite biter colony or not?
Yeah, so it's very rigorous, it's time consuming, and we produce colonies. So, for example, if we have two colonies that chew, let's say 50%, you can think of it as hierarchical, the selection. So we're looking at grooming and mite biting. So the colonies, for example, that chew the most mites out of, let's say, 100 colonies, we have about 120, 130 colonies that we use for selection. So we take the top performers and we'll cross the best to the best. So we'll take the best queen from, say, line A, and we'll produce drones from that queen, and then the best queens from line B will produce daughter queens from that colony. And then what we do is we instrumentally inseminate those, we'll cross them. So we produce breeder queens. And in each one of those breeder queens, their progeny are assessed. We assess the progeny and we test them. And then in some cases, they don't, for whatever reason, it's not the best cross. So that happens sometimes too. So bee breeding is constant work, but in many cases, there's also good crosses. So the good crosses are what drive the population forward. So you constantly have to select those. Bee breeding it's not so much a goal, it's a process. So you can't sit back, like we can't say, well, we got achieved 50% with this trait in 2015. We couldn't stop then and sit back and enjoy, all the hard work that we've done that's paid off and celebrate. Nope, you have to keep selecting. So you have to apply that pressure constantly. So that's what makes bee breeding difficult.
Right. So when you take a colony that you want to assess, are you still doing it the same way as you did in 2007, where the mites drop on a sticky board and you're looking at that board under a microscope?
Yeah, so that's what we do. We simply put a sticky board in, we'll spray it with vegetable oil like you would an iron skillet for frying your eggs. Not too much, not too little. And then 48 hours, we remove that board, we bring it into our lab, and then we have an undergrad, a field tech that will go through all the mites and determine how many mites are on the board. And then if there's, say, 200 mites on the board, we'll remove 100 mites and put on the glass slides. For years and years ago, we used to collect all the mites and put them, but that was too much work, too time consuming. So we take 100 and then we look at them under a microscope. They're all put on their backs, the mice are, so we can assess the damage easier. Then we look at the mites that are chewed, and then the ones that are chewed more, say for, if they have the shell chewed and all their legs missing, we kind of score that. So that would be a check would be put there in the box. So that might be a possible breeder if there's a large number of mice like that missing a lot of legs. But very often we'll just see 2 or 3 legs missing, or the pedipalps in the front.
So in your apiary, the colonies that are showing a good response to this behavior, they don't get treated, whereas the ones that are sort of struggling a bit, you still treat those. Is that correct?
That's correct. Yes, you have to, no matter what trait you're selecting for varroa resistance, you have some individuals, for whatever reason, still have problems. So they get overcome with virus, or there's a larger population of mites. Those colonies will be treated. So we typically will use like oxalic acid, works really well.
Mm-hmm. Right. And that's important because there could potentially be a hive nearby that it's bombing the rest of these colonies, and they won't be able to withstand it without being treated.
That's correct. Yeah, I've had it where we've had colonies we didn't treat, and the neighbors got infected, and they may have not have died, but they dwindled down, or they did die. And I've seen that quite a few times actually. So it's best to treat. The worst case scenario is to let everything, just let nature take over and let what lives, what dies. That's not the right approach. So you have some cases, you'll have colonies that for any trait, for whatever reason, they have a lot of mites and are able to deal with it, or there's no problem still. And then there's some cases where the mites are just not reproductive for whatever reason. She'll invade a cell, but she's not reproductive. And so there's a lot of finesse that's involved in breeding. And so you got to pay attention to all the details. But it's interesting to see those phenomenons where you have certain, extremes within a population, but yet they're fine. So,
right. So
we don't really answer for it.
Yeah, right. So how can people get involved in this program if they want to get involved, maybe try out some of these bees?
Yeah, here in the US and Canada also has got stock from us. When we started our breeding program, uh, we were just kind of working on the trait, fine tuning it. We didn't think anything was ready for prime time. And I think around 2012, 2013, we started having beekeepers come because we teach a queen ring course each year and an instrumental insemination course. And these beekeepers found out about what we were doing and they said, "Hey, you need to get this stock out." So, a long story short, a co-op was formed here in the U.S. where we had 7 states. They called the Heartland Honeybee Breeders Cooperative, and then in our state we have the Indiana Queen Breeders. So, in here in Indiana, they take bee breeding seriously. And as I mentioned before, we worked with our commercial beekeeper, and they've always supported us, which is great. So, mite biters are going to California every year, for example, being tested. And so basically a lot of the stock gets funneled out through these organizations, through the HHBBC and then the IQBA. But we also occasionally sell queens to other researchers. These are instrumentally inseminated queens because they're trying to improve stock also. And a lot of them are, going through the IQBA and the HHBBC. So every so often we'll get a beekeeper also that's wanting a breeder queen. It's just we don't have time to produce large numbers of queens and sell them. So we recommend they go through the IQBA, the Indiana Queen Breeders, because they have a list on there. I think there's like 18 bee or breeders on there now that have our stock also that they can get from them because we don't have time to produce queens and sell them.
Excellent. All right, let's talk about instrumental insemination and your business, Apis Engineering, which you co-own with your partner Dale. Interesting story how you got started there. So, if am I correct in saying that Dale came to do some instrumental insemination with you, and
Yes.
there wasn't enough equipment, so he said, "Hey, I can just make this stuff." Is that true?
Yeah, that story is true. That's it all. The genesis of Apis Engineering was it started. He took one of my instrumental insemination classes, and we got talking. He was a really great student, and I said, oh, Dale, I'd really like to have two more of these long bass type instruments. It's a European instrument. It's called a Schley instrument. So years ago, I used all Schley. So hey, I said, hey, I'd love to have another long bass instrument. And he's like, oh, I could whip you off a couple of those. In my mind, I was like, okay, we'll see. Because I've been told that before by other people where they brought me stuff and it was just complete. It's a nice attempt, but it was low quality.
You thought it might have been a fence post cut in half or something.
Yeah. So, because you need to have the finesse, everything. You need smooth movements. It's important. It has to be like butter. So 2 weeks later, he comes back with two of the bases, and I was floored. I didn't expect that. And then our friendship kind of spawned from that. And he later made the other components, and I still have those actually, through those two instruments he made. So they're kind of homages, to what's already out there. And then, yeah, through time, we got talking, and in my mind, I always was, one of my great people that I admire was the great Harry Laidlaw, who's considered the pioneer of honeybee genetics. Well, he also used to built his own instrument, so I always had this dream kind of being involved in or doing something like that. And then Dale came along, and that happened. So he does all the engineering, he builds the instrument. All I've done is give advice on, how it should be made or what works, what doesn't, tweaking the movements and things. And so in the end, we came out with, innovative products. And above all, like the Queen Station, for example, it's completely different than anything else. I mean, it's the most sophisticated instrument, I think, in the world
Mhm.
right now.
Yeah, and that's your flagship product. You've also got the micro station. Can you tell
Mhm.
listeners what's the difference between the two?
Yeah, the microstation is basically more It's a more economical instrument. It's you simply get an instrument and then you have to buy the light separate, all the CO2. So those components are separate from the instrument. Whereas the Queen Station, everything is on board. So you have one stand that the microscope's on, and then the CO2 unit is incorporated into that. So in the light system is also there. So everything you need is in one box. So it's
Yeah, and if people want to buy one of these, they can also go to you in Indiana and receive training. Is that correct?
yeah. So I teach instrumental insemination there every year. We have Dale and I. We have this year we're going to do 3 courses. And I think we have 4 or 5 spots already taken. So typically we have about 6 to 8 students a year.
Excellent. And I'm going to put the details about that business in the show notes for this episode. So if anyone wants to get some of this equipment, or if you want to get training, you can go along and have a look there. Is there anything else you'd like to have a chat about today, Crispin, before we wrap it up?
Yeah, I know recently you guys have got varroa mites, varroa destructor, and introduced to your country. And I think things that we've been doing here in the US can really help you guys out. One is importing germplasm. So like we can get permits, for example, to ship semen there and have it inseminated into queens and to help speed up the process of somewhat resistance. Because in the beginning, it's just going to be a tidal wave like it was here in the US, where there'll be a lot of mortality. But eventually it'll get back on track. But maybe if you can import some germplasm from other more varroa resistant areas, it might help slow that process down, instead of taking years for
Mm-hmm.
resistance, speed it up.
Mm-hmm. So just for listeners here, I'll explain what's currently happening in Australia. So it's been 2.5 years since we first saw varroa, or first detected it. And since then, it's pretty much taken over a small portion of one of the states, and it's been seen sporadically around other parts of that state. And we've also seen it in my state in a few areas. So it's probably taken over less than 10% of Australia so far. But at the moment, we tried to resist it in the beginning, but we're able to, and now it's kind of just marching along. Hasn't gotten to me yet, but it'll be here pretty soon.
Yeah, it's interesting to think about varroa destructor. Unfortunately, it doesn't seem to have any genetic incompatibilities, what I mean is they're highly inbred. So the varroa individuals that came to your country, maybe it was just, let's say it was 15 or 20. Within those individuals, because varroa mites, when she lays an egg, for example, it's the first egg that hatches is the male, and then 2 or 3 or 4 eggs, if she's really prolific, are female. So they mate with her brother and sister. So they don't seem to have any genetic bottlenecks like our honeybees do, which are haploid diploidy. But varroa is also haploid diploid, but they don't have that. So that's part of the problem with varroa too, is they can produce so quickly and be inbred.
Mm-hmm. Yeah, interesting. All right, Crispin. Well, thanks a lot for being on the show today.
Yeah, well, thanks for inviting me.